That's so weird. I show you. No, I'm
Just
God, do you have a napkin?
Maybe I'm trying not.
There's shit on the other side too.
What do you know about that? Yeah.
I think so.
Do you want me to like sound check, like log in and sound check it out?
Monitored on my phone.
Oh, because like it's electronic, huh?
It's actually pretty. So I actually yeah, so right now I just
Live now? It should be live. I'm gonna need my brother to call to see if
Yeah,
he
I
can
just sent Kelly the link. So it should be on his phone, but if you send
I know. Yeah, I'm excited. So next week, I'll start going
So you're gonna go.
they give you that I have yeah, so I
Is that one live? Okay. Should be.
well, central. I would have. The classes are in and they also require class. It's not do jobs. So I just didn't see it right. And they don't have an association. And I will kind of yeah, so it would most likely be something else, but Eastern. At least for this particular. So when does that start? August 5th.
I'm gonna go outside the door and do a sound check.
So I have there's so much so I'll be teaching photography 1, she'll be teaching photography 2. She's already been teaching photography. So she does something.
Okay, Ryan, this is your sound check. This is the sound check for Hannah's defense.
So whatever it is.
I think all these numbers right here.
Excited for it. Certainly not going Trying to see if I can unmirror image.
Oh, so that reverses the writing.
I can also send hard to vectors from the animations.
My blind. Yeah, it's just mirrored, so trying to fix that.
Welcome everyone, we're going to get started. It is a real pleasure to introduce Hannah George for her Dissertation Defense. I'd like to start by extending a very warm welcome to all of Hannah's colleagues, her friends, and her family, her mom and dad, all of her sibs, her sister-in-law, her cousin, and her grandparents, and many close friends who traveled quite a bit to be here today. So on this very happy occasion, so thank you for coming, and a warm welcome to you all. Hannah's during her graduate study was impactful in sort of the very best ways. She had impact on her field of study. That was through her exciting research. She had impact on her students. That was through her immense efforts and investments in her teaching. She had impact on our department. That was through her commitment to service and outreach. And she had impact on everyone she interacted with through her positivity, her enthusiasm, and her kindness. I asked Hannah's mother for some photos of Hannah. She was watching. This cute one was Hannah trying to be me. But she still comes off like super positive. Some of the other photos that she sent paint a lovely picture of Hannah's early interest in nature. Here's Hannah at just not even age 2, hugging her grandmother's flowers. Here she is dissecting the dogfish shark at age 10. And here she is posing next to a turtle on a Hawaiian beach. Looking at these pictures, you might think she was destined to be a botanist, maybe, a biologist, an anatomist. ecologist, a conservation biologist, but actually her true faith, and her mom pointed out this detail to me, actually lies in one of these photos too. But around her neck here is a magnifying lens, suggesting even at age 10 she was interested in this. So, and that's what she did. So Hannah's wet lab work is focused on small circular genetic elements called plasmids. These are passed from mother cells to their daughter cells. It's called vertical transmission, as well as between unrelated cells through a process called horizontal transmission. The study of these elements in our lab was a completely new path. And so Hannah was a true innovator with regards to this work. She voraciously read the literature, she learned basic techniques, she formulated computational models, she earned what's called a training grant, as well as several seed grants, and spent time with a world expert in plasmid biology, Dr. Ava Topp, at the University of Idaho, learning more advanced plasmid techniques. I think Ava might be on our live feed here as well. So I wanted to thank Ava and the Top Lab, and also Hannah's grandparents who fed and housed her while she was at the University of Idaho. As you'll see, some really exciting results, all the more so because of the pioneering efforts that went on behind this work. On the education research side, Hannah has worked closely with the just a world-class, amazing education research group that we are lucky enough to have here at University of Washington. She's worked on the effects of active learning in the classroom, also the effects of psychosocial intervention with regards to reduction of stereotype threat, how that may disproportionately aid students from underrepresented groups. And so this work can have serious impact on how we do education. And Hannah's right on the front lines of it, especially with regards to reducing the achievement gap in STEM disciplines. Her efforts in all of these areas were really remarkable. And to illustrate that, I'm going to use some photos from Hannah's love of sports and the outdoors. And I'm also apologizing to channel the spirit of Tom Daniel, and there's going to be some very bad puns. So here we go. So despite the stern consequences, sorry, Hannah would never bow out. She learned the rope and ended up, her PhD as a consequence ended up being very smooth sailing. She was never afraid to dive deeply into her. She gauged her pressure very well, and the last few months she had just a tremendous final ascent. A trailblazer, she moved mountains to get her work done, always exhibiting peak performance. But she never lost sight of the forest for the trees. As she was fishing for answers here, she became a true leader in the field. And her work for many has held just tremendous lure. In fact, many of my colleagues said that they thought her research was really flawed. With regards to the microbiology side, Hannah has truly mastered this plasmid rabbit. For those who aren't familiar, Hannah has the UW intramural tennis champion for 3 years. This is just what was going on, a small part of what was going on outside the lab. Inside the lab, she was also innovative in terms of sports. This is synchronized serial transfer. Here, I think she's gearing up for maybe lab part rouge. This one's actually real. This is our holiday party. We have a hands-free Jello auger eating contest. It's very serious, where we have to weigh the plates at the end to see before and after. And this year, Hannah and Ryan actually tied for first place. In all seriousness, Hannah's efforts in the lab represent a Herculean effort. Here she is with her collaborator Liv, posing in front of hundreds of plates. This is just a tiny fraction of what Hannah processed. Her dedication in the lab, combined with a boldness to explore new things, really led to some, I think, extraordinary results. So beyond the research accomplishments, beyond the sports titles, who is this Hannah Joy person? So she's someone pretty, as these pictures from our lab group show, you'll see in the middle of a lot of them is Hannah. She's just been a true friend to people in our group. She's so generous and kind to people, supportive and encouraging when needed. I want to recognize that kindness. She's really contributed to our lab culture and our department culture. And we'll really that. So thank you. Besides her kindness, she's also been tremendously persistent, as the shirts illustrate. In this photo, I just love. This is Hannah. I think her mom's quote. She just kept going always until she dropped. And here she is reading an animal picture book. Hand mid-grab into Cheerios, falling asleep. And this picture is actually a pretty good predictor of where she ended up even last week. This is sort of so just replace the Cheerios with coffee and the picture book with equals, and you got a good approximation. So again, it is a pleasure to welcome Hannah. As with many of my students, their titles turn out to be nice anagrams of describing them, and Hannah's title is no exception. So if you just rearrange the letters for Hannah's title, you get the following. Forsooth, do tell, if transcendent research becomes a sport, and she has no speed limit, and her interactions with in the lab, in the classroom, and with everybody in her life, she's just so adept at bringing people joy. So please join me in welcoming Hannah to bring a little joy to you with her research.
All right, thank you so much everyone for being here, and thanks to Ben for that amazing introduction. I'm really grateful to have all of your support here today. You all have made this experience one of the best of my life, and I just really appreciate that you're all here to see the very end of it. So, my dissertation title is called Of E. coli in Classroom, Stories of Persistence. And as you'll see, sort of as this slide indicates, that there are two parts to my research, both in the lab and in the classroom. And so my first two chapters that I'm going to be talking to you about, the big question is how do plasmids persist in bacterial populations? And we'll talk about plasmids more in a second, but that was, so that's mostly has to do with a lot of lab work, as Ben showed many stacks of plates, but also a little bit of mathematical modeling, which I'll touch on at the very end of telling you about my first two chapters. And then chapter 3, my question was more about how we can mitigate achievement gaps in the classroom and how can we improve learning in STEM. So to start with the plasmid side first, what are plasmids? So plasmids, here's a cartoon of a bacterial cell with its chromosome. And plasmids are extra chromosomal pieces of DNA, so they replicate independently of the chromosome, but they still contain genes that are relevant to the cell. And what's really interesting about plasmids is they can spread into other bacteria that are in the environment. So here's a plasmid-free cell, and the plasmid can cause the cell to create a pilus that connects to this bacteria, and you kind of creates a copy of the plasmid that can move into a cell that doesn't have that plasmid already. So in this way, plasmids can spread genes throughout bacterial populations, and not only just between bacteria of the same species, but they can also spread to other bacterial species that are in the environment. And this is the only real biological picture you'll see of plasmids and bacteria, but this is an example of bacteria conjugating that plasmid over to a different cell. So this process is called conjugation. And we also want to know where plasmids exist. So plasmids can exist anywhere that bacteria are, so they're fairly frequently found in bacterial populations, and they can especially be enriched in populations where there's antibiotics that are added to the system, because plasmids, they don't necessarily contain the core genes that are related to cellular processes, but they often have these accessory genes that allow the cell to be sort of convert sort of some advantage to the cell in certain environmental contexts. So when you have antibiotics in the system, they can sort of enrich these plasmids that might have antibiotic resistance genes for those antibiotics. And so we see enriched plasmids in places where there are livestock, or in plants, where pesticides or fertilizers are added to crops. We also see that plasmids can be enriched in medical settings, so in clinical settings, where antibiotics are often found. And we can often see plasmids moving between these two different areas that that humans come into contact with plasmids so frequently. So much so that a couple of years ago, the World Health Organization put out this recommendation that we should stop using antibiotics in livestock farms to prevent the spread of antibiotic resistant genes moving from environmental contexts into clinical settings. But just a couple of weeks ago, many of you read the New York Times expose on this company that was suggesting that livestock farmers should still give antibiotics to their feed indiscriminately so that you can prevent infections from arising, so to prevent the pig zero. And this is a problem, and this is not just a hypothetical situation that we talk about when we say, oh, plasmids can move from the environment into clinical settings. There was actually, a few years ago, you may have heard of what I like to call like the celebrity plasmid, so it's the most famous plasmid in the world, probably. But there was a plasmid that conferred resistance to colistin, an antibiotic of last resort that colistin is used for a lot of multivariate infections. And resistance to that colistin gene showed up on a plasmid in pig farms in China. And it then spread from China to Europe, to the US, and really all over the world. And even just this year, there was a paper that looked at Vietnamese, rural Vietnamese residents and their gut microbiome, and they found that 70% of the Vietnamese residents had this colistin resistance gene in their microbiota. So this really can spread all over the world, a single gene on a plasmid, because of this conjugation that can move genes on plasmids from one bacterial cell to another. So plasmid persistence is a big problem, and my research is this whole time been trying to look at how do plasmids persist in bacterial populations, and how does that spread happen, and why do they stay around? So to talk a little bit about plasmid dynamics, so it's kind of hard to see actually the gray cells that are around these plasmids here, but you can barely see. This is a population of bacterial cells. And if we want to see how plasmids are enriched or depleted in the environment, we have to think about cell growth and survival. So I just duplicated all the cells here, if you can see that. And I just did a sort of random duplication event of every cell. But if you have plasmids that are conferring an advantage in the environment, then they can be enriched faster. If plasmids are conferring a detrimental effect on the host cell, then you can have plasmids being reduced in the environment. So cell growth and survival, can increase or deplete plasmids depending. You also can have segregational loss. So this is the way that plasmids can be eliminated from the environment. So during a duplication event, like I just showed, you can have plasmids that don't segregate into both the daughter cells, and then it creates a plasmid-free cell with some probability. And you can also have plasmids being enriched in the population. You have to think about conjugation, of course, as we talked about before, when you can have plasmid-free cells that are incorporating new plasmids into those cells. So in this way, all these processes affect whether plasmids are existing in the environment or not, increasing or decreasing. So I just added, it's hard to see with all the lights, apologies with this, but there is, if we add an antibiotic to this system, so for example, I don't know if any of that shade too small. Okay, so let's imagine there is a red background that gets added to this environment that represents an antibiotic that the red plasmid confers resistance to. So in that case, you might have all the cells that didn't contain that red plasmid dying. And so when antibiotics are in the system, it's the way that conjugate plasmids can increase, or plasmids in general can increase, that confer resistance to that antibiotic. But it's still a big question, why are they persistent? So when you don't have antibiotics in the system, there's actually costs that are associated with carrying the plasmid. And so these costs might be because the plasmid is interfering with cellular processes, or it takes extra energy to express the genes that are on the plasmids. And so in general, in the plasmid literature, in the plasmid world, we say that plasmid-containing cells should be lost when there's no selection for that plasmid. They should get outcompeted by the plasmid-free cells because they have higher fitness. But if you imagine a red background going across the screen, so if you have an antibiotic that gets added that the plasmid confers resistance to, then actually the plasmid-free cell can die. The relationship changes, so now of course you have a plasmid-containing cell that are in the environment, and then There's still, though, even in this case, there's a question of, well, if the plasmids are costly, then we would still expect the genes, if they're beneficial, to eventually be incorporated into the chromosome so that the cell sort of saves on all of those costs that are associated with carrying the plasmid. And so this dichotomy of what it seems as if in environments where the plasmid is selected for or against, you still would never expect there to be the plasmid remaining. And so this is sort of in turn this idea that if plasmid-mediated genes are beneficial, then they should transition to the chromosome. That's sort of been termed the plasmid paradox, that in either environment you don't expect plasmids to be there. And so my dissertation, my question of how do plasmids persist, is really more of a question about what are the solutions to the plasmid paradox? How can plasmids sort of overcome this cost that's associated with them when there's no selection? And so my chapter 1, I look at an evolutionary solution and the consequences of that solution. And in chapter 2, I look at an ecological solution. And there are a couple other solutions that I explored during my PhD, but those you can ask me about later if you are curious. I'm just going to focus on these two for today. So first of all, I had to go and find a bunch of plasmids. And I always like to say that my fieldwork was done in Idaho. Some of you have heard me say that, but it was specifically done in Moscow, Idaho, here where that dot is, at the University of Idaho. And in this building, there's a freezer. And in this freezer, there is a shelf that houses many combinations of hosts and plasmids. And the owner of this freezer is the top lab. And as Ben was saying, I spent many months in the top lab learning from Ava Top and her group. And they really sort of adopted me as an honorary lab member, and I still attend their lab meetings through Skype and chat with Ava very frequently. And so I just also want to acknowledge right now that they've been a huge part of this, and all of the plasmid work could not have been done without the help of these individuals here. So I took all their plasmids, I brought them back into my freezer and the her freezer in Kincaid and now LFB. And I wanted to see all these plasmids and host combinations that I was able to gather from Ava's freezer, from many different labs she's collected them from. I wanted to see how persistent are plasmids if you grow them up where there's antibiotic present. So this is, I will mention whenever there's antibiotic present in the environment, but know that imagine there's shading of medicine. I'm going to stop saying imagine there's shading, but if there's antibiotic present, I will tell you. So if you grow up that plasmid containing cells, an antibiotic, all cells should have that plasmid. But if you transfer these every day in the absence of the antibiotic, then you will see sort of a drop of the plasmids if there are costs associated with those plasmids. And so in this way, we can measure how persistent a plasmid is in its host. And we sort of expect to see a graph like this. So we're looking at the proportion of plasmid-containing cells and how that decreases over time when there's no selection for the plasmid. And you're going to see a lot of graphs in a second that look like this. Is what the actual data looks like for one of those graphs. And you're looking at persistence profiles for two different plasmid host combinations, and they're both in E. coli, but they're two separate plasmids. And one, you can see the green one at the top that has higher stability or is more persistent than the one that's on the bottom. So as I transferred these over 8 days, the plasmids were dropping out at different rates. And so I did this with several other cells, or plasmid host combinations in E. coli, and you can see there's sort of variable persistence that's coming down. But there's also a few other categories of persistent plasmids that I was surprised to find, in that many are actually fully stable over this 8-day transfer period, or highly persistent, which sort of goes against that paradox that we think of when there's no selection for these plasmids, that they should be costly, but in this case, they don't seem to be. And I did the same thing with some of the plasmids from the top, and I put them into a Klebsiella pneumonia host, so a different bacterial cell. And you can see that, again, many of the plasmids remain highly persistent or fully persistent. I did this with, you got a third bacteria, so Shionella monodensis. And what I want to point out here is that I kept the colors consistent with the baseline being E. coli. So if a plasmid is fully persistent in E. coli in red, if you look at other red trajectories on the other hosts, that's the same plasmid, but maybe having a different persistence profile. So we can see from here, for example, one of the red lines, PALTS27, the plasmid, if you track that to the Klebsiella host or the Shionella host here, you can see that persistence is variable across the different hosts. So Surprisingly, yes, many were still stable, but there's still a fraction, certainly, that would fall under the plasmid paradox scenario in that they are costly, and how do these plasmids stick around? So we're still trying to answer this question of why plasmids can be persistent, sort of in this case on the right here, where you have plasmids being lost when there's no selection. And so you've already seen this graph where we measure persistence, and this is sort of, so you grow it up in antibiotic, and then you take away the antibiotic and see how it drops out. But it doesn't always have to remain a unstable situation. So one of my first hypotheses when I started exploring these ideas was that, and this has also been seen before in the literature, but if you evolve these plasmid host pairs in antibiotics, so imagine there is selection still here on the side, if you evolve them together under selection, and you're forcing that plasmid to remain in that cell, then after many generations, you might expect that when you rerun that persistence assay and say, how stable are these plasmids now? Well, you might get a less steep curve, so you might have more stability, simply because there are mutations along that trajectory as it evolved that allow the plasmid to persist more. And so I was particularly interested in what the effects of this host plasmid coevolution was on the plasmid persistence for these two different background hosts and plasmids. So we have E. coli hosts are always in rectangles, and Klebsiella pneumonia hosts are always in ovals. And I have two plasmids, P1 and P2 is what I'm calling them now. If you have questions more about the specifics of those later, you can ask me, but we'll be calling them P1 and P2. They both confer different antibiotic resistance effects on their host. And we can run the same thing here and say, okay, well, when we put the plasmids into these background hosts, we see a persistence profile that is relatively unstable. But I evolved all of these populations, replicates of these populations, for 400 generations. And you can see that all the replicates increase in persistence as they went. You can evolve bacteria and their plasmids together and see this increased persistence. And I will also say here that all of these graphs, every graph you'll see from here and from now on, there are at least double those graphs because I did the same thing with all of the reciprocals. And all of the results were very similar. But for ease of the presentation, I'm just showing one step. So the final, say that we have host plasmid coevolution, can increase plasmid persistence. So that's one solution to the plasmid paradox. But in particular, what's the effects of this? So imagine if we have two hosts with two different plasmids, and we can mix these together in combinations. And so previously, a lot of plasmid persistence has been done only on, as I've shown before, just an isogenic strain that you are tracking individually, and that hasn't really been looked at in populations very much. So in this case, we wanted to put two different plasmids with two different hosts together in one population. And here's the ancestral population we can imagine. And when you put them together, you can get this transfer of a plasmid into its neighbor host that has a different plasmid that confers a different resistance profile. And so this could create a multi-drug resistant cell in this occasion. And so in the ancestral group, though, we still expect plasmids are kind of not persisting, they're sort of dropping down in proportion of the population. And so you get sort of a small baseline of these double plasmid-contained cells, multi-drug resistant cells arising. But imagine then we do our experiment and we evolve in environment 1, we're evolving the P1 plasmid in its E. coli host, we're evolving the P2 plasmid in its Klebsiella host, and then imagine there's background colors here to show that they're evolving in their respective antibiotics. And then if we take away the antibiotic selection, and we can still have the same dynamic happening, but now these plasmids are much more persistent, they don't drop out of the population, and so you might expect that you get multiple events like this happening, and so you might see an increase in the amount of multi-drug resistant cells that are around. And so I'm going to show you what the results were of that by first explaining what the axes are and having you consider the graphs yourself. So when we put these things in co-cultures, we can look at the ancestral co-culture and take the evolved co-culture. And because, I'll start with the ancestral side, because you have two different cell types in there, you can imagine there's a bunch of Klebsiella cells that we're going to be tracking, a bunch of E. coli cells that we're going to be tracking, and each of those can have 4 different phenotypes, basically. You can have one that's resistant to its original red plasmid here with the Klebsiella. You can have one that's resistant to the blue antibiotic, resistant to the blue antibiotic that the plasmid has resistance genes to. And then you can have the multi-drug resistant cells that might arise, or plasmid-free cells. So we actually track 8 different cell types for E. coli and Klebsiella in the same tube. And so I want you guys to look at the data here and take just 10 seconds on your own to think about it. Okay, and now before I ask you to talk to yourselves, I just want to first say that this is giving you a difficult problem. It took me 4 years to figure out. So you guys have plenty of don't worry if about whatever you think about the graphs. But I want you guys to specifically focus on what is happening with the multi-drug resistant cells and talk to your neighbor about the main conclusions you make from these graphs, focusing on that purple trajectory. Go ahead, talk to me. Okay, let's come back together as a class or as an audience. So can I have a volunteer maybe tell the audience what you're talking about? What do you guys think is the conclusion with the purple cells? Yes, see.
You said there was a big difference between the E. coli host and the Klebsiella host.
Yes, absolutely. So that's definitely one of the main takeaways is that Klebsiella actually there's a lot more multi-drug resistant cells that are created in the Klebsiella type. And that could have something to do with the type of pili, how they connect to the background host, lots of different things. But yeah, we do see that there's definitely not as many multi-drug resistant cells in the E. coli. Any other thoughts? Thanks, Jake. Isa.
Having 2 plasmids is actually more beneficial because the highest one, base cell density is not the one with 2 plasmids.
Yeah, the highest density is not the one with the 2 plasmids, and that could definitely be because it's not as beneficial, so there might be a greater cost to having those 2 plasmids together. But it could also be that it takes longer to create those cells, so you have to have an event that happens, and so they're starting from no frequencies, and so they're increasing slightly. But yes, definitely there are fewer of those. What about between the ancestral and the evolved? My last question. Yeah, I can't see the lights back there. Yeah, Alexa, yeah.
There appears to be an increase in the amount of multi-drug resistant in the Klebsiella.
Yeah, absolutely. So when you go from an ancestral co-culture to an evolved, just like we talked about before in our cartoon examples, you do see a lot higher incidence of these multi-drug resistant cells. And this is a big message in the paper that we're writing about this project. And in particular, I also want to point out that this scale is cell density on the log scale. And so really the difference in purple cells here and the difference between number of purple cells here is much greater than it might seem by these graphs. So I'm just talking with Ben about this and about how this could sort of be represented if you're thinking about population size in cities and towns. And so my grandma is from a town with 600 people in it that's here, Clark Fork, Idaho. And if you consider that as being the ancestral co-culture that has that many double plasmid containing cells, versus Seattle, which has about 600,000 or a little bit more, that's how many cells are sort of represented here. The difference between a small 600-person town and Seattle. So really there's a big difference in cell numbers here. So great, yeah, so we know that you can have coevolution between a plasmid and its host, increasing the number of multi-drug resistant cells that are arising in the population. But we also were interested in, because you have these two different cell types together, and you're having a blue plasmid, P1 plasmid moving into a cell that's only ever seen a P2 red plasmid, then what happens with this plasmid stability when you're sort of moving into this new host? And so, for example, if you have the evolved P1 and P2 plasmids in their respective hosts, the Klebsiella loses its red plasmid, the blue plasmid conjugates into it. Now we have a novel evolved host for that plasmid. And so we wanted to look at how does the evolved cell do in that case when it has this novel plasmid that's incorporated into it. And you can see here, this is the Klebsiella host with the ancestral version, and it has the P1 plasmid. But here's the Klebsiella host that has never seen this P1 plasmid before, but as soon as we incorporate it into the cell, you see persistence is greatly increased. So not only did the host confer greater greater persistence in the host that evolved with its own plasmid, but also other plasmids that it encounters in the environment could also be more highly persistent. And we see this when we swap this as well, in that whenever you have a host that has never seen the plasmid before, it's evolved with another plasmid, then it can still confer increased persistence to new plasmids. So this is a sort of take-home message of why we have this sort of increased amount of multi-drug resistance across when you mix two different plasmid host combinations together. And finally, with this chapter, we also wanted to look at, once you have those multi-drug resistant cells, like think about our big world map. We don't want a lot of multi-drug resistant cells hanging out. So if you have those cells, how quickly are those lost? And so we have both an ancestral and evolved multi-plasmid containing cell, multi-drug resistant cell. And to lose those plasmids if the host is no longer resistant to any antibiotics, you first have to become single plasmid containing cells and then finding a plasmid-free cell that's now sensitive to the antibiotics again. And so I looked at every single one of these transitions here to see how does the persistence change. And we can see if we're just, first I'll say that the co-evolved plasmid that this host evolved with was the red one, so the blue one is the novel plasmid that it's encountering. And so I'll show you a bunch of persistence graphs. Here's again the proportion of plasmid-containing cells on the y-axis and transfer over 9 days. And so here is the first transition that we've measured. We've talked about this before, is that when you have the ancestral version with a single plasmid-containing cell becoming a plasmid-free cell. In the ancestral version, it drops out quickly. And then in the evolved version, it is much more stable. And then we can also look at the reciprocal here, so where you have the blue plasmid being lost, how quickly does that happen in the ancestral versus the evolved? And you can see that also, as you've seen in the previous slide, that also happens much more. The drop is much more quick in the ancestral version. And then if you look at the double plasmid containing cells now, and you're saying, well, okay, so how quickly is this red cell lost? This red cell lost so that there's only the blue cell left. And in order to do this, we actually have to maintain that blue cell to get at this. So there's a caveat that this stability actually is happening in the background. This blue background. So the background of the antibiotic that's selected for that single plasmid. But we're tracking in this graph the double plasmid containing cells. How quickly do they drop out from this by tracking this red plasmid to that trajectory? And we can see that in the case of evolved versus ancestral, the evolved drops out much less quickly. And then the other alternate one is the alternate direction is also true, where with this there's that red background here, and so you have the evolved version is dropping out much less quickly than the ancestral when you're tracking the blue cells, how quickly they're dropping to just becoming red cells. And so in this case, we know all these different transitions happen much more quickly in the ancestral, and indeed when we measure this across, take away all the antibiotics, they're just measuring how do the double plasmid containing cells drop out in the evolved versus the ancestral. We see that indeed, again, the multi-drug resistance is dropping out much more quickly in the ancestral version than the evolved. So that brings us to wrapping up my longest chapter, chapter 1. So what there is an evolutionary solution to the plasmid paradox, and that when you co-evolve the host and its plasmid together, you can see increased plasmid persistence in the absence of selection, and you would expect it to drop out. And the consequences to that are that host plasmid, the co-evolution of a host and its plasmid, that can lead to the emergence of multi-drug resistance at a greater frequency. And so I'm briefly going to touch too on a second solution to the plasmid paradox of how you could have these constant plasmids maintained in bacterial populations. And this is more of an ecological solution. And I just want to really quickly show you again this slide just to remind you that we expect, without any selection for the plasmid, that there is costs associated with the plasmid. They have less fitness than those cells that have no plasmids. And then when you add, if you're in an environment that is selects for the plasmid and there are genes that confer benefit to the host, then you would expect that the plasmid genes may be better if they're incorporated to the chromosome for the fitness of the cell. But also, for this experiment, I actually looked at a plasmid that is non-conjugative here to sort of mimic this host chromosome incorporated gene to compare the differences between plasmids that are conjugative versus non-conjugative, because a lot of the costs are associated with conjugation in plasmids in the first place. So we can still sort of have a good idea of why conjugative plasmids persist by comparing these two conjugative and non-conjugative types. And so this is actually a mathematical model that we use to simulate the graphs that you'll see on the next slide. I'm not going to talk about this at all. I just wanted to communicate that that's what is behind the results in this part of my chapter. But in this, we have three different players that we're sort of tracking, as I explained last time: conjugative plasmids, non-conjugative plasmids, and plasmid-free cells. And we can see that in constant environments, you can get antibiotic, or when there's no antibiotic in the environment that's selected for the plasmid, of course you'll get the plasmid-free cell increasing in frequency faster and be maintained and the others are displaced, because the plasmids are costly in that situation. If the antibiotic is present that is selected for the plasmid related genes, then you would still expect the plasmid that's non-conjugative, that doesn't have that cost associated with conjugation, taking over and not allowing conjugative plasmids to persist. But our solution is that maybe if you alternate selection in the environment, then you can get maintenance of all three of these different types. And that's specifically because there can be an advantage associated with conjugation itself. And so if you look at one of these periods in which we're moving back and forth between antibiotic-free, antibiotic-present, antibiotic-free, when there's no antibiotic, the black line is increasing. And then as soon as we transition into an antibiotic environment, then that line decreases. But there are many plasmid-free cells in here that the conjugative plasmid can actually move into. And so that increases with a lot greater frequency than the red plasmid, which can't move those plasmid-related genes into this sort of reservoir that's available for the conjugative plasmids. And so in this case, we can get conjugative plasmids, even though they may be more costly than non-conjugative plasmids, or as proxy, what is the same genes in the chromosome that can't conjugate. And we see, even interestingly, if you extend the period length and wait long enough for the red cells to die out, that they can never reestablish. So you can actually just get maintenance of the conjugative types with these alternating selection environments without evolution occurring at all, just alternating the environment. So that's to wrap up by chapter 2. So costly conjugative plasmids can persist when you alternate selection. And now I'm going to transition to the other sort of half of my experience here at UW. And my question really is, how can we contribute to higher and more equitable student persistence in STEM? And there's a number of ways that I've explored that might contribute to this. But first, I just want to say a big shout out to Bird that looks at these questions of how do we increase student learning, persistence, and sense of belonging, et cetera, all these things that we want to increase for students. But they're always keeping an eye on these gaps that might exist between certain groups of students and others. And so it's really important to think about these gaps and what's causing the gaps. And so when I'm talking about a gap, what I mean and how we measure it in our research is that we're looking at gaps controlling for academic ability. So if you have a student who comes into your classroom that has one GPA and identical to another, they should be performing at the same baseline. But sometimes we see that students underperform where we expect them to, and that means that something in our classroom is happening. And that could be really due to like me as the instructor, I'm not doing something pedagogically correct, or it could be the subject itself, and that sometimes STEM disciplines are considered more difficult for others, and that can create this sort of psychological pressure on students. And so I also am interested in what can mitigate these gaps that might come up, and returning to sort of that psychological pressure that sometimes exists in the classroom for certain groups of students. I'm looking at how stereotype threat, how that might affect student performance in Biology 180, and how we can perhaps mitigate that. And that is one of my dissertation chapters focus. And first, I'll talk about what stereotype threat is. So it's sort of the fear that you're going to confirm a stereotype that's associated with your own group that you identify with. And that can be any group that anyone identifies with. And so who's affected by this? So you can have women who are sometimes stereotypically not supposed to be as good at math or science as men. And so that can sort of bring up, they can stereotypes associated with gender, or first generation college students across disciplines, they might experience stereotype threat because they made the radio stereotype, they don't belong in college. Same with underrepresented minority students across disciplines, we see the stereotype threat affects these students. Even in the elderly, when they think they're doing memory tasks and they're told that they're old, they might experience this stereotype threat, the effects of it. White men in athletics can, if they're told that that you're measuring athletic prowess, they can actually, that stereotype can affect their performance. And so all of these groups their performance in different tasks can be affected. And how does it manifest? And here's a very classic paper that first talked about stereotype threat as a thing, but we look at sort of how is performance affected by stereotypes that might be made salient in your environment. And so usually it's separated, the graphs that you see in many of these studies are separated into groups where you're experiencing or expecting them not to experience the stereotype, and then students who may be experiencing the stereotype associated with the task at hand. And so in this case, we're looking at mean GRE verbal items that are solved. And this group was told that the study was measuring personal factors involved in problem solving, and this group was told they were just measuring psychological factors. And when they were told that personal factors were measured, Black students did worse on that exercise than Black students who were just told that it was psychological factors. So when you make sort of a stereotype potentially salient to students, that can really impact performance. And this is a very well-established phenomenon across many, many studies. And what the main question that I had coming into this project is, can we sort of mitigate the effects of stereotype threat in Bio 180 specifically, but on academic performance in general? And it's sort of based on this paper that I read as an undergraduate, actually. This is Cohen et al. In 2006, published in Science, and they did this intervention. This is a group of middle school students that they're looking at their average assignment performance across a term, and this is like the term broken into different performance blocks. And the students are given this intervention at the very beginning of this term, and their performance is tracked over time. And you have the European Americans are this line right here, so they start out with higher performance than the African American groups. But the control group of African Americans who didn't get this intervention, they continue to do worse throughout the term. But those students who got the intervention, those Black students who got the intervention, their performance stayed stable. And so I read about this intervention and wanted to try it out on our students in Bio 180. And so first I'm going to have you all take 10 seconds and do the activity yourself. So think about this for 10 seconds to yourself and pick one. Okay, 2 or 3, but I want you to turn to your neighbor and talk to your neighbor about why those values are important to you. Okay, let's get back together as a community. And I know there's many wonderful conversations going on right now, but due to time, I'm going to stop you from talking about that. Feel free to pick back up again after the presentation. So basically, you guys just did the activity that we asked the 180 students to do, and that this researcher asked these middle school students to do. And there's a couple extra questions, but this is the main bulk of it. And students are not asked to talk to each other about it, they're asked to do it online, writing a few paragraphs about why they think that these values are important to them. And so we did this in Bio 180. I started this project when I was still an undergrad a long time ago with these three individuals here, and we all read the same paper, and we all said, that's so cool, that can happen for middle school students, can we make it happen for our biology 180 students? And then we went to Scott, and we said, Scott, we're going to do this. And he was like, okay. Well, he just said, okay, right away. And so we did. And then we also got my co-first doctor, Sarah Eddy, in on the project too, after we sort of got it started. But we did this, there's 550 students per section in these courses. They're high structured courses, so there's lots of active learning going on. We did it across these 3 years. This is when we collected the data after preliminary studies of a couple years of collecting data. So these are the years that we actually had everything working. So we had 2,383 students in the study, and we had this proportion of different groups. And we were particularly interested in looking at what the effects of this intervention was on females or underrepresented minority students who might be experiencing stereotypes in our classes we were expecting. And again, these are done online, not with your neighbor. We randomly distributed them during weeks 1 and 2, so students did this twice during the quarter. And we had a treatment and a control. And the control is really interesting in that it's the exact same activity, but students are asked about 3 values that are least important to them, and they have to talk about why they're important to someone else. So they still see all of these same words, they talk about them, but they're not relating them to themselves. And so that is our control, and that's the standard control for this values affirmation exercise. And our results were, first of all, our outcome variable is exam performance. So all of the exams that the students take during the course are added up, and we're looking at how the treatment affects that exam performance. We use linear regression models to control for certain aspects of student background and test for the effects of different backgrounds on the performance. And so it's important to remember that when we are looking at this graph about to pop up, that the data is controlling for the cumulative GPA of students, what they bring into the course as their GPA, in the section that the students are in. And so here's what we see. We have percent exam points earned, and this is URM females, and underrepresented minority females, underrepresented minority males, white females, and white males. And in the control group is in red, and the treatment group is in blue. And these percentages are giving you the percentage difference between exam performance in the control and the treatment group. So our biggest increase was with URM males, who saw a 4.2% increase in their overall exam performance when they had this treatment that they were given. And so just to wrap up, we did see that there was an achievement gap in exam scores between underrepresented minority and white students in biology 180 across these 3 different years. And we saw that values affirmation can reduce this gap. And I also here want to take a moment to thank John Parks, who helped us administer this to all of the students. But more importantly, after we gave him the results, he emailed me the very next quarter and said, I assume we're doing this in 180 for however long we're going to continue. So it is still being used every quarter in 180. And I really want to thank John Parks for I didn't know John Parks has just been so accommodating with any experiments that I want to try. So that's just an extra added thank you. And now I'm going to go through other thank yous to all of you. So first, I want to thank the Kerr Lab that I am so fortunate to be a member of. And I definitely want to thank my advisor, Ben. I know many of you have been on the receiving end of his excitement for whatever research you're talking about. But I have to say that it's really something extra special when he's your advisor, and his whole true emotional investment is in you and in your success. And Ben was the one who came to me about a year into my PhD and said, I know you're really interested in education. Would you like to make education a chapter in your thesis? And that was just a really big deal for someone who did want to go into that, and just to have that support from Ben was really impactful. He's also created a wonderful environment for us all to learn and grow together and have fun together. And I really want to thank Kate Dickinson here for just being a wonderful lab manager. I don't know how the Kerr Lab would run without you. And so she is a force of nature, as many of you have experienced in this department. So big thanks to Katie for doing everything that she does for our lab. Also, big thanks to our undergrads. So we had an undergrad appreciation event in the Kerr Lab last week. And these undergrads in the background are just really have a big impact on our lab. And especially thanks to Riley, Leo, Shannon, and Jessica, who were all 4 undergrads that worked particularly on the plasma top projects with me. Also, from the very first weekend I joined the Kerr Lab, we did our annual canoeing trip that only was one. But there are always the greatest intentions for us all to get together, and here is my first buddy in the Kerr Lab, Sarah Hammerlin, who was back at the time, and making our agar Jello plates, as Ben mentioned when we do our competitions. But there are many wonderful traditions in the Kerr Lab that make it a great place to be a student. And I also want to thank just the latest round of Kerr grads, because you've all been so instrumental in my happiness and my professional, where I reached professionally. And so thank you to all of you. I also want to thank the Top Lab, who I hope the camera will be for all of you. So thank you, Top Lab. But especially Ava Top, who, like I said, has just treated me as one of her own students. And anytime I have questions or anything, I can Slack her, and she responds immediately. And she is just has a world of knowledge at her fingertips for plasma biology. And anytime you want to know anything that's happened in the literature, just ask her and she'll know. And so I really just appreciate how much she's welcomed me as part of her lab, and including the 6 months that I've actually spent in her lab working on plasma-related things. And I also want to thank these 2 postdocs, so Thibaut and Wesley. When I first came to the Top Lab, they taught me everything that I know about technical aspects of working with plasmids, and they are both excellent collaborators and also excellent friends, and really allowed me to integrate myself into that department when I was there and spending so much time in Idaho. Really, and the whole grad community there is just really amazing, and I consider them a very special component of my grad family. Bird, I want to thank all of you, the lecturers that have really done so much to make me the researcher that I am and ask the questions that I want to ask. All 3 of these have been really instrumental in sort of allowing me to do the things I want to do in the department and being encouraging. Special thanks to Mary, Pat, and Allison, who, when I showed up as a first year, wanting to be involved in Bird, they just started showing up to their lab meetings. They really welcomed me and have been huge supporters my whole time here. I really, really appreciate them. Brian, I couldn't find a picture. But I do want to give a special shout out to Brian. So not only is he one of the Bird members that is really contributes a lot to my own development, but he was the first person who taught me how to be a science mentee. So he also ran the HHMI undergraduate program when I did my first lab experience as an undergraduate. And I had no idea what science was. No one in my family is trained scientists. And I joined that group, and Brian told us what it was to tell people what you do in 3 minutes in an elevator, and how to write grants and talk to PIs, and how to communicate to your PI so that you are not a bad mentee. And so maybe Ben should also thank you. So very, very big thanks to Brian. Also Ben Wiggins. So he has been so confident in my abilities to just hop into a bird-related project that he's invited me to be on a couple of his projects, and I'm really, really grateful. And also for all of his help in the department as a whole to gain everything. He's given me a lot of opportunities, and I really appreciate that. Ellie, she is sort of my partner in crime in the Bird group since we were both graduate students early on, and she's still there. And she has also given me so many opportunities to work on bird-related projects, and it's just a great friend as well. So thank you, Ellie. And then Jake, of course, which I'll get to you more later. But also, we have had many collaborations together with Berg, and really, I appreciate all the conversations that we have about teaching all the time as we sit next to each other in our desks and have for the last 6 years. And then, of course, Scott. So Scott has given me so many opportunities and amazing ways of interacting with Berg, and also just the confidence of the scientist. He taught my first biology class when I came to UW, and really had a big impact on me going into the field at all, and going to education as well. So I peer TA'd with Scott when I was an undergrad, and that was what originally when I started, I was like, oh, I'll never become a teacher. That was what I thought. And then I started working with Scott, and he took so much time to help us understand what it was to be a scholar in the field of teaching, and really made a difference in my trajectory. And then, of course, all of the graduate students that have talked about education with me, have participated in breakfast club mornings where we talk about education pedagogy, and that have taken Scott's 505B class and spent a year working on the meta-analysis that Scott and Ellie are working so hard on right now. And it's been amazing to have this community of grad students who are so invested in teaching, and I just really appreciate all of them. This is us at 8 o'clock in the morning, coding all together a bunch of educational papers. So that shows you the dedication that exists in the department. The copy and the bagels might help too. So thanks to Scott for that too. I want to thank my committee members. Carl Borstman and Beth Traxler, in addition to Scott Freeman and Kirk, are on my committee, and thank you so much for your support during all of these 5 years and helped steer me into the projects that I wanted to go into, and also especially for supporting my chapter in education and allowing me to do that. I definitely forgot people on this list, but the biology staff and the GDPC, the graduate program committee, all of their support allows this department to run, and that's my science to get done. And thanks, special thanks to the custodians and Amine who really make this also an environment that is a welcoming place to be doing science. All of these groups, I'm not going to explain all of you, but you know who you are. And yeah, I just appreciate all of you. Funding, Beacon, and the NSF, Department of Biology, and NIH has allowed me to do a lot of the plasmid work that I've done. So that is very, I'm very grateful for that. I've also definitely missed a lot of you. This is just a smattering of activities that the grad program students have done and that I've been able to get involved in. And the community here of grad students is really phenomenal. It makes UW Biology a very special home to be a part of. And if I don't have you up here, you know that you're special too. All special thanks too to the Darlers. So every quarter we do an intramural sport in the department, and this is just 3 of the probably 12 different quarters at the least that we've done different sports. But especially shout out to whoever designed these shirts many years ago, and we all still proudly wear them. And also special thanks too to my ultimate pursuit team. A few of my teammates are here, and they have also been with me through this entire process as I've been a student and have allowed me to sort of do things outside of the department that are also fun. So thanks for showing up, guys. Before I start off, these next 3 people, in true Curlab fashion, I randomly selected which 3 of you were going to go first, second, and third, because it was causing me too much stress to pick one of you go first. So Jake got the first stick, and these 3 people are my best friends in the department and the world. They've just made my life so much better. I'm so grateful to all of them. And so Jake has been at my left shoulder my whole grad career, right next to me in his desk. And I think we both know each and then when we should ask if that's an interesting thing to talk about, or when we should let them continue doing their work. So we really have worked well together and are always excited to tell each other about the exciting things that we think about every single hour of the day. Sorry, that's an alternate co-host to us. We're in our office that was annoying. But I will say that we've done many ventures both outside of the classroom, but also most importantly is just that sort of desk area of the office. This is big texting us when Eastway and I rock on one of our adventures, saying that he missed both of us in the office. But really, it's made coming to work every day a joy. Like I always look forward to coming to work and coming in on Mondays because I know that my best friend's sitting right next to me. Liv, you have come into my life in the last couple of years, but you are definitely one of those friends for me, and we have done so much in the lab together, and I like to think that yes, we are synchronized. When somebody calls our name, we're just like, hi. So I will say that in the last year, Liv has really helped with a lot of my class projects. I would not have been able to finish up everything I did without her co-projecting with. And I'll also say in the last year, I've gotten to be a much, much better experimental biologist because of Liv. Like, she is the most rigorous scientist I've ever worked with. And I know she's going to go super far, but she's definitely has made my science much better. And I want to thank her for that. And to say I'm looking forward to the rest of your career trajectory. So no, it's going to be great. And also we're roommates, so we were still come home and still talk, and we're still best friends. So I think that also says something to that. Eastway, you have just been like my emotional rock in grad school, and we've had so many adventures, both around Seattle, when you were also my roommate in our new apartment that we moved into, and cleaned top to bottom and made our home. And also, she was always one that was like, you just still have to travel, you still have to do other things outside of research. So she dragged me to the Galapagos and to Ecuador to buy some jungles, and to her home in Mexico, where she invited us so graciously, along with other very dear friends of ours, to share in her family Christmas down there, and also to Iceland just last year. So she's taking me all over the globe, and even when we're not in these places, she still writes me little notes so that I remember how awesome all of those places that we go are, so that I remain happy even when I am in Seattle, which obviously I still remain pretty happy in Seattle. But thank you, Eastway. Your friendship has really meant a lot. Family. So I definitely am also missing lots of people here. These are some of my closest family. A lot of them are listening from afar. I want to specifically point out my Uncle Kelly, who is probably listening right now, but also, despite not being trained as a scientist, has watched every single one of my evolution lectures that I've ever given on Panopta. And calls me occasionally every couple of weeks when I'm teaching a class and say, well, I didn't understand this. Could you explain it all? Is planning on watching all of my biology lectures that I'm going to be teaching this summer. So I look forward to learning it along with Kelly still. And he's just been a really good confidant over the years too. And all of these people are so special and have done so much. I want to have a special thank you to my grandparents, Karen and Mike, who are both here. They're the grandparents that I stayed with in Idaho, and so I spent 6 months with them too while I was in Idaho. I wake up in the morning, my grandma would have coffee ready, breakfast, a lunch, out the door. And she and my grandpa, they would get up and have coffee with me every morning, and my grandma is a retired professor from the University of Idaho. She's the only PhD in my family. And the advice that she would always give throughout the school process has been really impactful. And I thank her for being interested in what I do and for knowing what this experience is like and talking to me about that. And my grandpa too is a very special person. And I really appreciate, I'm so grateful that they live in Idaho and Moscow, and that Ava also lives there, so that I can spend so much time with them. I want to thank my brother Nick, who is my buddy, and he has been my buddy, especially in the last year. We go to get dinner once a week, every Tuesday, and he's really been a big support throughout this last year as I've gotten ready for my PhD defense. I'm moving in age order, but I'm including here my cousin Amanda, because she is like a sister to me. We grew up together and we're the same age, and we just graduated together this year too. So, so I went to her graduation in Missoula, Montana, just earlier this month, and now she is here at mine, and she probably knows more about my life than any of you in here. She's my closest confidant and knows most of my secrets. And then my older brother Ethan, who we have been together through everything. We've had so many adventures together, and I just appreciate you more than you'll ever know. And I also appreciate you bringing in my sister-in-law, Karen, to the family. And you also are like a sister to me, and I thank you for your support throughout this whole process. And always asking me if I wanted her to bring me anything this last week, which is very appreciated. My older sister Chelsea, who's here from Ohio, and she has back in Ohio my wonderful brother-in-law and my nephew Wyatt, but she came here today to be with me Dissertation Defense, and as an older sister, she's just been there for me always, and I really appreciate that. And then of course my parents, who supported me through everything. And I just want you to know how much I appreciate that. I know you for that, but I do. So, that's it.
We're a little over time, but we do want to give Hannah a chance to answer a few questions if people have.
So I didn't understand any of that, so.
You can talk about later.
The whole 180 though.
Yeah. Yeah, John.
I wonder if you have hypotheses about what's changed about the host cells during your coalition of plasmids that makes them more hospitable.
Yeah, well, great question. So what you actually don't know is that I sequenced everything, all the evolved replicates, and Liv has been really helpful in putting that data together, as well as has Tebow back in Idaho. But yeah, we actually have many of the plasmids themselves that did change and had higher persistence. They dropped things like the conjugative apparatus that might be especially costly, and they also, some lost really big resistance genes, like extra resistance genes that they had. Some of the plasmids didn't change at all, and there were mostly changes in the host. So there's a variety, because we had 6 different replicates, evolved replicates, for 4 different combinations, the E. coli with P1, E. coli with P2, Klebsiella P1, Klebsiella P2, so we did 6 replicates of all of those things. And so it's very variable, but things like that are happening in the plasmids. But we're interested actually in pursuing that question more, to see what that's actually going. Thanks. Yeah.
So to follow up on that, in general, is there a theme? I mean, do they tend to be big deletions, or do they tend to be changes in regulatory features on the plasmid or changes themselves?
On the plasmids?
Yeah, I mean, because one might hypothesize that making big deletions would be advantageous as opposed to just changing regulators, for example. So what was there a theme that you saw?
Yeah, so it's really this interestingly in all of the evolved replicates that have like these 2 resistant genes they have, it's actually like the same big deletion of these genes across many replicates. So they've all evolved for 400 generations in their own population, and they all have this same section of the gene large deletion taken out. And then the same with some of the plasmids that have the conjugate apparatus taken out. Some of them have sections of it, and others have like many of the TRA genes that were removed. So it's big deletion flows. We didn't have any SNPs actually in any of the plasmids. Yeah, here.
Do you know how vitamins feel about the extraction? Either students who are taking the class and don't know what it's about, or students who are taking the class and figure it out, or people about that?
Right, so my undergrads that have worked with me in the lab, I've actually asked them, because after they've done the series, sometimes the education research will get brought up, and they'll want to know what I do, and I was like, well, did you ever take that exercise back in the day? And they're like, oh yeah, like, that, I do remember that, but it was with so many other things that they have to take, so many different surveys, they didn't really think much of it, and they were like, oh yeah, I just did it, and then that was it. There was no, like, animosity towards it, or like, puzzle, like no one was puzzled by it. They're just like, oh yeah, I did that. It's another one of the things I did. But yeah, when we did the actual experiment and some students got the control exercise, there was, I remember talking to a few students and they're like, yeah, the control is a little weird, like asking me like what other people thought. Was there like important? Yeah, Jack, thanks, Mary.
Phil, Paul, what do you think the mechanism is that's driving that change? So they take this assessment and identify is that changing it? Help me close the gap.
Yeah, so one of the big sort of mechanisms of how stereotype threat is sort of thought to affect performance is that it's taking up sort of your working memory if you have some sort of stereotypes in the environment. And so even if you're not thinking that if you're not thinking about it, like you might be thinking about it subconsciously or not, but that working memory or sort of you're trying to reconcile what's going on in your environment with your own beliefs. And so when you take that value of affirmation intervention, it sort of gives you a booster a little bit is how it's thought to of as, in that you're like, well, there are things outside of this this arena that are important to me and that I care about and I'm good at. And so that sort of boosts them against this stereotype that might be pervasive in the environment. Yeah, great. Yeah,
so following up on that, I know that stereotype threat and values affirmation has gotten some flack, some heat with the literature and the community for not being reliably replicated. Can you what are some hypotheses that you have about why it might be replicable in both in some settings or even in the same setting that we have?
Yeah, great question. So absolutely, we sort of view that exercise as not being like a silver bullet that you just give this to students and stereotype that it's gone. So there are a lot of variable results across studies for values affirmation, and sometimes they never see any difference at all, sometimes they do. I think it really depends on the environment. So if there is, maybe there's a really good classroom culture where you're not even affected by, like stereotype threat isn't really made as salient, so that it might not be something that you were worried about, so the values affirmation doesn't work because there's nothing to sort of alleviate. I know we did it over the 3 different classes that we did it, 3 different years that we did it in. One year there was a huge difference, and then the next year there was not a difference, but there was also, there was no gap at all. And so then the 3rd year there was a very almost significant difference. So even across the 3 years that we did it, we pulled all of the data, but across those 3 years there was also variability. So I think it really depends on the environment that you're at, the students that you're working with, and sort of what's happened leading up to that activity in your classroom. But yeah.
Do one last.
Yeah, Mike.
Would the plasmid research have any application for drug companies? Because it seems like all the evolutions were done under the duress. Would companies be able to look at that and all they develop drugs on the movement of plasmids, or would they?
Yeah, no, that's a great question. So really, we often prescribe multiple antibiotics, and so this is sort of if these, like when you prescribe cocktail drugs, for example, and there are plasmids in that environment that have both of those, like are resistant to both of those antibiotics, then this sort of gives us an idea and clinicians an idea of sort of how you might expect that antibiotic resistance to go away once you take away the drugs that they're giving patients. So it could possibly feed into some of that. But yeah, in general, this is kind of all bad news in a way that plasmids are more persistent and antibiotic resistance can be maintained in environments because for us, we're trying to think of how can we minimize the amount of plasmids that are in the environments, the ones that minimize the antibiotic resistance that gets spread through the environment. And so this is sort of like it's really a problem. And now that we know it's a problem, then we need other people, other researchers to sort of think, how can we deal with this?
I noticed on the interventions, the one group that did more poorly were the white women.
Yeah.
Was there any
Yes, so it actually increased slightly the gender gap when we did the activity. So there was a significant increase in the difference between men and women on the test. But women overall, like that was a very small change, and like so that it was not significant, that change, but between the white women and who took the intervention or not. But across all of the women, there was a slight difference. They actually across the women, it was there was no difference if you took the intervention, but it actually widened the gap a little bit. So that is a side effect that happened, a small side effect. It was a small amount.
So we're going to end it there, but for those who are interested, we will have a gathering somewhere in the neighborhood of about 45 minutes in the fifth floor of Elsby. Let's thank Hannah for a very
I saw you scream white
He and I have to go to
Okay, that's fine.
Oh, don't people want to
Do you have change for 20?
Yeah.
Or 2 10s? And then I'm gonna grab my shoes.
Bunch of organized cash. There you go, 22.
Yeah.
Give you singles, but I'm trying to avoid that. Oh, here's the pen.
Sweet. Thank you.
Thank you. And the box is right there if you want to hear.